if we've built a cell, our job is going to need to be able to pinch it in two, okay? And so we want to make something that goes from there to sort of a constricted version where we can have, DNA on this side and DNA on that side, and really ultimately physically separate these two cells. And so we're going to do that during cytokinesis, where we're actually going to break them in parts. So we're going to physically separate the cells. And the way that we're going to do that is we're going to actually have to build something that pinches it down. And so this is much larger. We kind of need a rubber band again, but a massive rubber band made of a lot of different components. And so we're going to actually build in a ring. And this is going to be called the cytokinetic furrow. It is a structure that's built to be able to constrict the cells. It needs to be able to break things in two. So if you imagine here is the spindle as it's elongating, holding on to the pieces of DNA. We want to make sure that we create this pair of molecular scissors, this ring, this furrow, so that it really doesn't entrap things there, that really separates them off. And so there is initially microtubules. They are going to contact each other. It's this point at which they contact each other that provides a signal for where you're going to build this. And so it's going to find the middle of that spindle structure, and it's going to build that thing there. This furrow is made of actin. So as much as the key players thus far in cell division and chromosome segregation we've discussed about have been microtubule-based primarily, the way that you're going to create this cell shape change is through actin. And so you have this basically actin ring, also with the action of myosin, a key motor. And in the same way we briefly talked about muscle cells using actin and myosin to contract and get closer together, myosin in this case with other factors can act to really just bring these rings and constrict them so that you have a much smaller structure there until you finally get to the point that you can pinch off. Okay? And so then we're going to be able to actually make a structure that comes down and puts the membrane so close that we can reorganize it, really distinguish and fully separate off these two daughter cells.